All right. So each time a cell reproduces, it divides into 2 new cells. When each of the new cells divide, the result is 4 new cells. If this continues, how many cells will be present after the cell reproduces? 6 different times. So basically, this looks like what type of growth? What do you guys think? Exponential or logistic? Exponential. So that's that J-shaped curve. Let's see what you guys came up with. See if you got the right number. Huh. We'll talk about that. Oh, I like 64. 64. Okay. I don't know. You go from 1 to having 2 to 2 to having 4 to 8 to 16, 16 to, and then if you double that for the 6th time, you get 64, okay? So that's like cell growth. That's one type of cells growing, one way that cells grow. Should have been 64. Okay. So today we're going to look at how cells divide and why they divide. We'll talk about DNA and how it's packaged in the nucleus. We'll talk about how cells prepare for cellular division and why it's all, that whole preparation to divide is just as important as the dividing itself. You guys all remember we copy DNA onto RNA, wiggles through goes out into the, to find a, to make a protein by assembling and forming. Yes. Come get one. I did that at the very beginning of the hour. Yep, that's okay. So as the body of a multicellular organism grows larger, its cells do not also grow large. Instead, the body grows by producing more cells. Let me ask you this question. Why do cells not just get bigger to make the body get bigger? Why? Why do cells make more of themselves? Why don't they just blow up like balloons? Yes, put that in, what you just said. I should have 32 of you answering. Well, close to it. 28, something like that. So new cells are needed to help tissues and organs grow. So why don't cells just expand? Let's see what you put. They are easier to maintain, yep. Ah, there's the kicker. The bigger the cell gets, the smaller the surface area to volume ratio is. So that whole surface area to volume ratio that a few of you put there is what we're looking for. If a cell gets too big, that surface area to volume ratio gets too small, and it can't bring in enough nutrients to support what's inside of the cell. So as old cells die, new cells are going to take their place, and new cells are going to also be there to replace damaged cells. So cells, they reproduce for many different reasons. They divide for a lot of reasons. First one is surface area to volume ratio. Second one is they get wore out. You have to replace them. Okay? You bleed, yeah. I don't know why I just repeated what you said because that one didn't fit, but yes, you bleed sometimes if you get cut. I think I'm on autopilot today. My brain just took off and I went with it. So a cell grows larger by building more cell products, right? Some cell products might be proteins, might be organelles, might be Golgi apparatus, might be chloroplasts, might be mitochondria, might be endoplasmic reticulum. But the cell must take in more nutrients, process them, and get rid of waste. All of that making of cell products requires energy and nutrition. A cell's ability to exchange substances is going to be limited by that surface area to volume ratio. And as the cell gets larger, substances must travel farther to reach where they are needed. So cells get so big and they have a choice. They either stop growing, they die, or they divide. So the work of cells is done by proteins. And as the cell gets larger, the more proteins are required to maintain its function. We all know proteins are made of amino acids, right? And if the cell gets too large, DNA instructions cannot be copied quickly enough to make the proteins that the cell needs to support itself. You guys hear about the cell that wouldn't share? It was selfish, you know. I know. I just realized I told that one before. Did I tell you about the famous cell? It was a celebrity. Right? So the cell size is also limited by the cell's DNA. Our DNA contains over 30,000 different genes in it. That's a lot of genes. Mason, you got a lot of shoes, but I don't think anybody's got 30,000 shoes. Now imagine 30,000 genes. Yeah. The really fancy ones are called designer genes. That's like Mickey Minaj's DNA, right? No? What is it? Nicki Minaj? What is it? I don't know. P. Diddy's genes? I don't know. What? You like that? What's her name? Mickey Mouse? Is it Nikki or is it Mickey? Nikki. Okay, I don't know. I get to be old. I don't have to know these things, right? And she's never listening to Mickey, whoever. So each daughter cell has a higher surface area to volume ratio than its parent. Why does each daughter cell have a higher surface area to volume ratio than the parent? Hold on, I can't get my thing working here. There we are. Why does each daughter cell have a higher surface area to volume ratio than the parent? Final answer in 5, 4, 3, 2, 0.5, 2.75. Oh, you guys aren't participating like you should be. There's 2 of them. Okay, they're younger. I don't know. Ah, smaller right there. Ding, ding, ding. They're actually half as small, right? Why are they half as small? Because they're half the parent, right? That's where they came from. They cut the parent in 2. They're there, right? Okay. Yeah, they'll grow again. They got a G1 phase, right? You guys remember drawing those out? So each new cell also contains an entire copy of the cell's DNA. If I'm going to make a new cell, I better have all the instructions on how to make more of everything that cell needs, right? So I got to have a whole extra set of DNA. So because larger cells are more difficult to maintain, cells divide when they grow to a certain size. Your cells get so big, and they go, oh, I'm so big, it's time to divide, and they divide and make more of themselves. Just like clone yourself, make another you. I don't know if the world needs another me. What do you think? I always tell my biotech kids, I will clone myself one day when it's legal. I want a little me running around. It's on my bucket list. Yeah, teach them how to step on your daughter's boyfriend's toes. Yes. No. She did. They can't find him. I don't know what happened to him. So the large molecule of DNA is organized into hereditary units called genes. Okay? So imagine this collection of genes. Okay? It's like I don't know what the gene store is, but it'd be like having all the different genes out there. So a gene is a segment of DNA that codes for RNA and protein. We copy DNA onto, RNA wiggles through it, and goes out into to make a protein by assembling and forming. Now here's the thing. When we copy DNA onto RNA, we copy one gene at a time. Think of this. You have in your body, in each of your cells, more than 30,000 genes. Each gene tells you how to make one protein. So we copy DNA onto one gene at a time. That means each set of RNA has the instructions on how to make one gene or one protein. Now think about how much smaller RNA is now than DNA. It's 1/30,000th the information. Small. Okay. So DNA is going to be organized and packaged into chromosomes. Everybody say chromosome. How many chromosomes do we have? Anybody know? Everybody say 46. 23 pairs. Everybody say 23 pairs equals 46 chromosomes. So a prokaryotic cell has a single circular molecule of DNA. We have, think in your head right now, and take your finger like this. Everybody put your finger in there and draw 46 lines. Okay, this is taking longer than I thought. So imagine 46 lines in front of you in this little make-believe piece of paper. That's a lot, right? Now take both fingers and draw a loop. That's pretty simple, right? That's the difference between us and bacterial DNA. Bacteria is a loop. We are 46 lines. Now, until I told you that, how many of you thought our DNA was simply this very long structure that was in every one of our cells? Yeah, like that. You see that model of DNA, right? We all been told this is DNA our whole life, right? So here's DNA. It's this very long structure. It's found in every one of our cells. It contains all of the blueprints for life. Blah, 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 boring, blah, right? We've all heard that. Bohooey. It is 46 lines of that stuff inside of every one of our cells. And it's actually 92. We'll talk more about that in a minute. Okay? We'll talk about it. So I'm awaiting responses for 2, 4, 6, 8, 10, 12, 13 of you. Come on now, get on the ball. Or on the Peardeck, one or the other. All right. So this loop of DNA contains thousands of genes. We contain over 30,000. They just a few thousand. We are way more complex than bacteria are. That's no surprise. But it's important to understand why. They have one looped chromosome. We have 46 different chromosomes. So eukaryotic cells contain many more genes arranged on several linear DNA molecules. Make sure you're also taking notes. Eukaryotic DNA is going to be wound into highly condensed chromosome structures with the help of many proteins. And the DNA and proteins make up a substance called chromatin. Everybody say loose chromatin, wind around histones to make chromosomes. So the first level of packaging is done by a class of proteins called histones. This is a group that you'll actually collect 8 of these proteins together, and it makes a core, and then we start to wind our DNA around it. The long DNA molecule winds around a series of histone cores in a regular manner and is what is called a nucleosome. And under an electron microscope, this level of packaging resembles beads on a string. We'll talk about that in a minute. And that string of nucleosomes lines up in a spiral to form a cord that is 30 millionths of a millimeter long. Very small. So small you cannot see it with that microscope in front of you. You need to bounce electrons off it and have a computer make an image in an electron microscope to see that. Now, during most of the cell's life, its chromosomes exist as coiled or uncoiled nucleosomes. So that means most of the time we will not see chromosomes. Most of the time, if I look through that light microscope, I just see a nucleus, and I can't tell if there's a chromosome there or not. Do you think if I wound DNA tight enough, I'd be able to see it with that microscope in front of you? Yes. Here's the thing. If I have you guys all run out in Canada, and I spread this classroom out all over Canada, and I fly in a plane, would I be able to pick you out of the woods? What if we all stood in one spot? Maybe, right? Maybe. Okay. So as the cell prepares to divide, the chromosomes condense even further, ensuring that extremely long DNA molecules do not get tangled up during cell division. So why do we make chromosomes? To organize our genetic information. Imagine having the pages of your book just kept in your backpack and not bound in a binding. And then let's say I put 2 books worth in there and ask you to separate them. Oh, that's not going to happen, right? Okay. It could, but very unlikely. How about, is it going to happen every day without mistakes? No. Less than 3 hours? Probably not. So, let's make DNA yellow. Little strands of DNA. Let's just pretend there's 46 of them there. Let's make our histones red. And remember, look, you guys might want to draw this. This is called chromatin. It could be. It's okay. That's DNA. This loose stuff is what is called, I'm sorry, the loose stuff is DNA. This stuff here is called histones. And this stuff winds tight. So as it winds up, you actually get 1, 2, 3, 4, 5, 6, 7, 8 histones together. I'm just going to make it simple here. I'm going to put just these bundles, but just know there's 8 histones inside of each of these bundles. And what happens is the DNA winds around it, and it makes these structures that under a microscope look like, that's a terrible drawing, look like little beads. And these are what are called nucleosomes. And if we take it one step further, this keeps winding tighter and tighter and tighter, okay, until I end up with this thing called a chromosome. But a chromosome actually contains 2 things. It contains 2 of these structures. Each structure referred to as a chromatid. And each chromatid is held together in the middle by a tight area that pinches them together. That is called a centromere. Centromere. That means that these 2 together are actually 1 chromosome. Is this a pair of chromosomes here? No, it's a pair of chromatids. So if I'm going to have a pair of chromosomes, that means I need what? 4 chromatids. Terrible drawing, but I hear you. Now let me ask you this. How many chromatids are in each pair of chromosomes? How many chromatids are in each pair of chromosomes? There's 4. Each chromosome contains 2 chromatids. So if you have 23 pairs of chromosomes, how many chromatids do you have, really? There's actually 92. Yeah. 23 times 2 times 2. Yeah. So, did you guys get that? This is where biology teachers have taught this wrong for years. It happened to me. we have loose material called chromatin, which contains DNA and histones. It winds tightly to make these nucleosome structures, and then it supercoils to form chromatids. There's 2 chromatids in each chromosome, and we have 23 pairs of chromosomes. You guys get that? Not easy. But the thing to understand is that each pair of chromosomes contain 2 chromatids. That's one chromosome. This is another chromosome, each held together by a centromere. Are chromatids identical, like clones of each other? Yes. Are the pairs of chromosomes clones to each other? Everybody say no. I have a brown-eye gene, and I have a blue-eye gene. We'll talk about this in Chapter 12 more. Okay? But they code for both eye color, let's say, but not necessarily the same eye color. But these 2 chromatids would be for blue eyes, both. These 2 would be for, like, brown eyes, both. You get that? Okay. That's melanin. This is another protein. You have a different color melanin. That's all. So the nucleosome cord forms loops that are attached to a protein scaffold, and those loop domains then coil into the final, most highly condensed form of the chromosome. Now, many dense loops of chromatin form the rod-shaped structures that can be seen in a regular light microscope, and we will look at onion root tip cells going through mitosis probably next Tuesday, something like that, maybe Monday. Now, each of the 2 thick strands of a fully condensed duplicated chromosome are called a chromatid. How many chromatids are in a chromosome? 2. How many pairs of chromosomes do we have in the human body? 23, which gives us a total of 46 chromosomes, or 92 chromatids, right? Boo. It's not fun, is it? Or is it? That's fun, right? What? Tin? Tid? Yeah, I know, right? Tin? Tid? Yeah. So each chromatid is made of a single long molecule of DNA. You think I'd have it figured out 12 and a half years in, right? Yeah. Halfway there. I don't. I'll be here until I'm 80, right? So identical pairs called sister chromatids are going to be held together in the middle by a centromere. What? Yeah, he is. 70. 71, I think. So during the cell division, the sister chromatids are separated at the centromere, and one ends up in each daughter cell. Each new cell has the same genetic information as the parent cell. This is why my skin today is hopefully the same skin tomorrow. I don't want skin cancer, right? That's a good thing. Now, you want most of your cells to be the same tomorrow as they were today. An exception of that would be like sex cells. You would not want all of your sex cells to be the same. Otherwise, we would all look the same, and we wouldn't have any variety. We need biodiversity, right? So there are times when you want your DNA to change and mix. There are times when you want it to be the same. The first type of division we're going to talk about is called mitosis. That's where everything stays the same. And in Chapter 11, we're going to talk about meiosis, which is where you make sperm and egg. Yes? Which one? If it's meiosis, sperm and egg, I might ask you to hold off till Chapter 11. Oh, that's different. That's genetic, where they actually do choose the genes they want. Can I explain that to you at the end? I promise I will, though. I promise. That's a longer explanation. So all new cells are produced by the division of pre-existing cells. That's the cell theory. Cells come from other cells. And the process of cell division involves more than cutting a cell into 2 pieces. You first have to go through the process of making more of the cell, dividing your DNA, and preparing to divide. So each cell must have all of the equipment needed to stay alive. All newly formed cells require DNA, so from before that cell divides, a copy of DNA is going to be made for each new daughter cell. Remember I had you do the little cell pie chart yesterday, and there was the S phase for replicating DNA? So each new cell will function in the same way as the cells that they replace, hopefully. If not, something has gone wrong. Prokaryotes, we're talking about bacteria, have different DNA. They have that circular DNA, and it's attached to the inner cell membrane. And the cytoplasm is divided when the new cell membrane forms between the two DNA copies. Meanwhile, the cell continues to grow until it doubles in size. Why would it have to double in size? Because it's going to split in two, right? Why would you split in two? Because your surface area to volume ratio gets too small, because you got too big. And it looks something like this. This is called binary fission. Bi means two. Fission means to separate or cut apart. Okay? I thought I missed something. It felt like I did. So the cell is constricted in the middle like a long balloon being squeezed in the center. This is, of course, still talking about binary fission. And eventually the dividing prokaryote is pinched into two independent daughter cells, each of which has its own circular DNA molecule. So the DNA, then the cytoplasm divide. We do it the same way, except we have a process we go through. We first divide our DNA, then our nucleus, then our cytoplasm. That make sense? So this is simple prokaryotic bacterial division, binary fission. The prokaryotic cell copies its DNA, makes two loops, then it separates its cytoplasm between the two loops, and you have two new cells. There's a high degree of error in here, and there's not a nucleus to worry about. That's good and bad. A lot of mistakes can happen because there's no nucleus to protect the DNA, but it happens a lot faster. How much faster? Our DNA divides about once a day. Or I'm sorry, our cells divide about once a day. Bacterial DNA from one cell, you can end up with millions of billions in one day. Depends on the bacteria, but anywhere between 1 minute and 5 minutes, yes. Okay? Usually a lot quicker than 5. So imagine exponential growth, how many you would end up with in a whole day's worth of a few minutes, right? It's literally millions of billions. Way more efficient than us, but they have a higher degree of error. Why? It doesn't matter, because they're one cell. We make a mistake in dividing our skin cells, we get cancer. Big difference. And eukaryotes, the reproduction in eukaryotic cells is more complex. Eukaryotic cells have more organelles. We have organelles, right? We have mitochondria, chloroplast, Golgi apparatus, endoplasmic reticulum, a nucleus, all of that stuff that we've been talking about in previous chapters. And the DNA within the nucleus must also be copied and stored and separated. We have more information. We have to catalog it into chromosomes, right? 92 chromatids, 23 pairs of 46 chromosomes. Ooh, that's a lot of info. 30,000 genes, 2.2 billion base pairs in there. Quick summary, I'll let you work on your reviewing keys. Because larger cells are more difficult to maintain, cells divide when they grow to a certain size due to surface area to volume ratio. Many proteins help package eukaryotic DNA into highly condensed chromosomal structures. Why chromosomes? The same reason you organize your pages into a book, to organize it, so you don't lose it. If you lose, let's say, a chromosome, or if you have an extra chromosome, you can end up with something like Down syndrome. And all newly formed cells require DNA, so before a cell divides, a copy of its DNA is made for each daughter cell. Remember, each daughter cell is going to have to be its own cell.